First one is if you have no plans over lunch and are near a computer, I highly recommend listening to this talk. It's just a streamed online seminar that we have from our friends at UCLA. Brian Drucker, hopefully most of you know Brian Drucker or who he is. He's at OHSU. He's probably the most likely person in Oregon I can think of to get the Nobel Prize. Invented Gleevec and has been really important in terms of developing cancer-specific drugs. And so it should be a really fascinating talk. We should also have a really fascinating talk in our biology department seminar, and that's tomorrow at noon. Kim Beatty from OHSU is a relatively new employee there. I don't know if she's going to get the Nobel Prize. We'll have to see. She's come up with some really fascinating tools for working with particularly mycobacteria and some of the pathogenic bacteria which are out there. So now we'll shift over to molecular biology, which again, a lot of the stuff that Brian Drucker does is really very closely connected to molecular biology and its understanding of molecular biology, particularly chromosome rearrangements, which is why he was able to develop some of these drugs. So we'll maybe talk a little bit about some of that today, not a huge amount. So again, today is the last lecture with new material. There'll be a review on Friday, midterm on Monday. Questions about that at this point? I will have regular office hours, and then if you have questions, email me, or we can also set up extra office hours outside of my regular ones, because I know a number of you have genetics right after this. Okay, so with that, let's move on to everybody's favorite part of lecture, which are clicker questions. So first question, the formation of gene families are probably due to gene duplication, purifying selection, a molecular clock, gene conversion, or mitotic recombination. Thirty seconds. Ten. Okay, everyone seems to agree on this one. I like that. Formation of gene families is, rather than are, probably due to gene duplication. Purifying selection, actually, if anything, would get rid of those gene families. Molecular clock is great for doing phylogeny. Gene conversion is actually, again, moving away from gene families because you're going to be getting back one copy. And mitotic recombination was something I thought up because I was trying to come up with another answer to fit on these things. So our next question is, the most common transposons in the human genome are autonomous DNA transposons, non-autonomous DNA transposons, autonomous LTR retrotransposons, non-autonomous LTR retrotransposons, non-LTR retrotransposons. Bah, 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 bah. 30 seconds. Ten. Okay, most people seem to think non-LTR retrotransposons. What would those be? It's the name that we use for them. Signs and lines. So long interspersed nuclear elements, short interspersed nuclear elements. By far and away, these are the most common of the transposons that we have in the human genome. Autonomous DNA transposons are very common in bacteria, but actually not very common in eukaryotic systems. We do have them. Non-autonomous DNA transposons were the first ones that were found by Barbara McClintock. Autonomous LTR retrotransposons are basically retroviruses that don't have any capsid proteins that are left with them anymore, but they're only about 8% of those, let's say 8% of our genome is those particular kinds. Non-autonomous LTR retrotransposons, there actually aren't very many of those in our genome. We didn't really talk about them. And one of the keys, I guess, to this question is what's autonomous versus non-autonomous? So yeah, so autonomous code for everything that they need to move. So if you're a DNA transposon, that transposon encodes the transposase. If you're a retrotransposon, then you have a reverse transcriptase and either an integrase, which is what you have with the LTR transposons, or an endonuclease, which is what you have with these non-LTR retrotransposons, which again are the most common. And it turns out that the signs are actually even more common than the lines, probably because they're parasites. These are the non-autonomous, non-LTR retrotransposons, but there wasn't enough space on the slide to put that whole thing in. So our last quicker question for today. Chloroplasts are inherited like mitochondria, so they are inherited cytoplasmically, inherited equally from both parents, always recombinant, always present in two forms, never lost. Thirty seconds. Ten. Oops, let's look at these results. I like it when everyone seems to agree, and I agree with them too. Yeah, so they're just inherited cytoplasmically. They're not inherited equally from both parents because that wouldn't be what you normally have from cytoplasmic inheritance. Always recombinant. There's very little recombination that happens between these different genomes because they're not going through meiosis and lining up with each other. Always present in two forms, not particularly. Never lost. That has nothing to do with inheritance. But turns out there are mechanisms to make sure that you end up with at least one mitochondria or chloroplast in the next generation. But again, we haven't talked about that. So talked about last time were the protein families. And Transposons, again, most of what we talked about with these transposons are the DNA transposons, which again are what you get in plants and bacteria. They're really useful tools, as we talked about towards the end of the lecture. But in our genome, most everything are these retrotransposons. And we spent more time talking about the LTR retrotransposons because they're like viruses. But also we understand a lot more about how these guys are actually replicating. And so that's why we spend more time talking about it. Very briefly talked about some of these organelle genomes. And then I've got my red italics down here at the end, which means we didn't talk about it, so I'm not going to be testing on it. Are questions about that? Actually, no, before I move on. Okay, today we're going to talk about eukaryotic chromatin. Move pretty quickly through structure and nucleosomes. Hopefully most of this is review for you. 30 nanometer filaments and 300 nanometer filaments. This is really how the structure of eukaryotic DNA is inside the cell most of the time. Metaphase chromosomes are a very specific case of extreme chromosome compaction. And then we'll talk a little bit about heterochromatin versus euchromatin. And the real key to heterochromatin versus euchromatin is that heterochromatin is basically inactive chromatin, and it's really inactive because it's been compacted. And so this whole idea about compaction of DNA is really important for whether it's active or inactive. A lot of that has to do with the so-called histone code, and this could really be histone modification code. And we'll talk about all the modifications that are happening, or I should say some of the modifications that are happening in histones. And then if we get to telomeres, we'll talk about telomeres, a problem that you have with linear genomes, but then we'll go from there. Yeah?
Before you start, so how can there be a mitochondrial parent, like you left off lecture last time?
Oh, how can there be a mitochondrial parent and separate from a nuclear parent?
Yeah, where does that come from?
Okay, so the question here is, sorry, repeating it for people who had to leave. Yeah, how can you have a mitochondrial parent who's not one of the nuclear parents? You have to do that in a so-called in vitro experiment. So you actually will enucleate a cell, so take the nucleus out, and so that just has the mitochondria, which in a mammalian system have all come from the mother. And then you put in a new nucleus from two different nuclear parents. And that's exactly what Metalipov and his group out at the Primate Center have done and shown that this works. And in fact, you can then correct mitochondrial diseases that way.
What about
What about chimeras? I'm not sure I quite understand your question. In some ways, that is a chimeric, would be a chimeric organism because it's come from various different sources if you think about it from that point of view. Well, if you have an embryonic stem cell that then develops into a full organism, yes, that would be a chimeric organism at that point. And that's in fact exactly what they did. It didn't develop fully in an organism, but it actually went through development, some development. Yeah, interesting ethical question to think about. Again, well beyond the scope of this course. So really, we've got pretty much two kinds of chromatin. There's either chromatin that's not very well compacted or chromatin that is compacted. Always, this all comes back to the problem that DNA is really long for the amount of material which it actually encodes. You have to compact it in order to get it inside a cell, but at the same time, you still have to have lots of proteins that are going to interact with that DNA. And so if you've compacted it, it still has to be accessible. And so that's where you have a lot of difficulties in terms of understanding this whole situation. So just purifying DNA from eukaryotic cells, you basically get two kinds, either this so-called beads on a string, these blobs with a little string between, that's a nucleosome with DNA between it, or under normal salt conditions, it's usually this 30 nanometer filament, which, as you can see here, is a really highly defined blob. And so we'll look at what we think that blob actually looks like as we move along. In the cell cycle, most of the time in interphase, you have these not ridiculously condensed DNA. These are interphase chromosomes. Most of these are going to be 30 nanometer filaments or some modification thereof. You have replication, which of course means you've got to at least somehow deal with some of this compaction because the proteins have to get in there, then the massive amount of condensation you get in M phase, the metaphase chromosome, that's when you can actually see chromosomes under the microscope, a regular light microscope. And it's the only time you can actually see chromosomes under a light microscope, is when they've been completely compacted in this mitotic state. Then, of course, you have cell division and things move along. So if you think about chromosomes in just a very general sense, forget what they're coding to start with. They really need three things, and this is at least for linear chromosomes. They have to have replication origins, and it's usually more than one. You have to have some way of separating your DNAs, and that's the centromere, and we'll talk a little bit more about that later on. And then if you've got a linear DNA, you have this problem with ends, and that has to do with the way that DNA is replicated with leading and lagging strands. The way that organisms with linear genomes deal with this is this so-called telomere structure that we'll talk about. So replication organs, again, clear you need for replication. Centromeres you need for cell division, and telomeres to deal with the ends of your genome. So that's all they really need for any given chromosome. So let's look at these. In an interphase cell, most of your DNA is complexed in this form. Some of it is complexed again in this beads on a string form. Now it turns out that this bead on a string form is the easiest to work with, but again, it's by nowhere near enough compaction that you would need to get everything inside the cell. And even this image here, this interphase image, is after having broken down a lot of protein DNA interactions. The cell doesn't really look like this. This is about a 1,000-fold compaction. If you move all the way to a metaphase chromosome, this is about a 10,000-fold compaction. But because you can isolate this particular form of DNA, and it's the relatively small piece to look at, people actually understand probably the most about this particular form of DNA. And it turns out this is your active form of DNA where most proteins can interact with it. Also known as the beads on a string, and again, it's just because the beads on a string are what you expect and you see when you look at this. So if you take a preparation of DNA like this and treat it with a very non-specific nuclease, and nucleases, what do? They chop up DNA, and this is an endonuclease, which does this, so it chops in the middle of your DNA. The key to this experiment is use very, very small amounts of it for very short periods of time. And so basically what it does is it will chew up DNA where it's not being protected by a protein being bound to it. And so that particular experiment will give you, when you separate these DNAs on an agarose gel, just separating based on size, you'll have most of your DNA is relatively small, but still about 150 nucleotides. But because you have really small amounts, you're actually going to have 150 nucleotides approximately down here at the bottom. Then you'll have 300, 450, etc. And what that means is you've cut between each of these various different nucleosomes. And so what that tells you is that the histones are protecting about 150 bases of DNA. You can purify these DNAs and the proteins that are associated with them. If you now treat with one of these nucleases for much longer, you see you've got 8 proteins, and those are the 8 histone proteins which are forming the nucleosome. If you add a linker histone, which is Histone H1, then you have a very similar kind of pattern here when you treat with micrococcal nuclease, but it's about 170 base pairs as opposed to 150. And here we've got 147 base pairs, 168 base pairs. That's the case for humans. Varies a little bit from species to species. So now if we look in a little bit more detail at what this nucleosome structure looks like, one of the reasons we know so much about nucleosomes is we actually have high-resolution structural data for these nucleosomes. And that was published in 1997. At that point, it was the largest crystal structure which had ever been determined. We've gotten beyond that now. But here you can see the approximately 150 base pairs of double-stranded DNA wrapped around these 8 proteins present here in the middle. These 8 proteins in the middle are the so-called core histones. They're H2A, H2B, H3, and H4. In this image, H2A and H2B are orange and red, and H3 and H4 are blue and green. So you can see here, hopefully, if you're not colorblind like my brother, you have two dimers of each of these. So you've got a dimer of H3 and H4 here, a dimer of H3 and H4 here. A little harder to see, but there's a dimer of H2A down here and a dimer of H2A, H2B down here. So there are 4 sets of dimers which are forming this. These proteins are among the most highly conserved sequences that anyone's ever looked at. And what that means is, well, we've had DNA around for a really long time. Compacting DNA is something that cells have had to do for a really long time. And the actual sequence of your protein, exactly what those amino acid residues are, is probably really important, or at least it's been selected for really long periods of time for being very, very similar kinds of sequences. That being said, these histones, when you purify them out of cells, have multiple different modifications. And these are all covalent modifications that happen to the amino acid side chains. And we'll look at those a little bit more later on. So if we now take each of those individual histones and look at them, kind of H2A, H2B, H3, and H4, they're all really similar in their structure right in the middle of the proteins. There are these 3 alpha helices which fold up into this structure. It's also known as the histone fold. It's a nice motif. It's not stable by itself. It's only stable when it's complexed in a dimer with another histone. And you can see that right here. Here are these 2 which are associating to form a dimer. And you can actually probably pretty reasonably guess that these are parts of a gene family. It turns out that these sequences in and of themselves are also quite closely related to each other. The differences between all of these different histones have to do with their so-called tails. So this is the piece of your amino acid sequence which is not in the histone fold, either N-terminal or C-terminal relative to this central fold domain. The tails here is really where the action, if you want to call it, is, certainly in terms of histone modifications. Almost all of them are happening here in these tail structures and not as much in this histone fold. And it gets not surprising because the histone folds all have to interact with the other histones. It's these tails which are going to be interacting with the DNA and then other proteins which are present there. If you just look at the sequences, and again, I mentioned that all 4 of the people call them canonical histones, the standard histones, are related to each other, so they're part of a gene family. They also have ridiculously high amounts of positively charged amino acids in them. 20 to 25% positively charged amino acids, which is not terribly surprising because what are they binding to? DNA. And what's the charge of DNA? Negative. So again, not terribly surprising that that's the case. And these are, just because of seeing all these charges, gives you a pretty good idea of what these histones are interacting with. They're interacting with the phosphates on the backbone. They're not interacting with the base pairs. If you look at, again, the structure of a nucleosome, we talked about these tails already. If you just look at the structure of the nucleosome, all these tails actually really do hang out on the outside of the nucleosome. They're not directly interacting with the DNA there. How do we know that? Well, now you do the opposite of that experiment where you did the nuclease to chop up the DNA that's not associated with protein. Now use a protease to chop up the protein that's not associated with DNA. And if you do that, you end up chopping off all of these tails here. So this is the experiment which shows that these are clearly projecting away from the nucleosome. Other experiments you can do, you can now take these histones without their tails, put them together with DNA, and they will actually make a nucleosome. That's what's shown over here on the side. And if you've taken off those tails, it turns out that nucleosome is much less stable. So yes, they're hanging out on the outside of the nucleosome, but they still serve to stabilize the nucleosome as well. So let's look at this nucleosome again in a little bit more detail. We've got same color coding. Blue and green are histone H3 and H4. Yellow and red are H2A and H2B. They come together as this octamer. The DNA wraps around them. DNA wraps around basically as these two loops that go around. It's actually not quite two complete loops, but it's very close to it. And you can see that here is one part where there's not one piece here. But one of the neat things about nucleosomes, and again, speaks to selection that you've had for probably billions of years, is that these nucleosomes are basically identical to each other, flipped over one way or the other. And what that means is you can actually pack nucleosomes on top of each other. And it doesn't matter if you pack nucleosomes on top of each other with them facing in one direction or facing in the opposite direction. So stacking nucleosomes is relatively easy to do. And also it means that if you look at the DNA that's associated with these nucleosomes, the DNA that's not bound to that core particle is pretty close to each other. So these two ends right here, the beginning and the end of your nucleosome, are really quite close to each other. If we zoom in now and look a little bit more at how these histones are interacting with the DNA, again, not surprisingly, most of these are long positively charged side chains. These are the ones here which are in red. But above and beyond the electrostatic interactions that you have with the DNA, you also have multiple hydrogen bonds, about 150 hydrogen bonds in a core nucleosome DNA interaction. So basically, any way that you could possibly think of to have DNA interacting with protein, that's what's happening in terms of this nucleosome process. But again, it's almost all happening at the backbone or in a non-specific kind of interaction, because if you had really specific nucleosomes, then they're going to have lots and lots of different lots of different histones. You're going to have to bind all over the DNA, because those are going to have to bind to those very specific sequences. So if you want to have something which is relatively efficient, you want to have something that binds really well, but really well non-specifically to DNA. Well, that's not completely true. It turns out that you can actually get some specificity of binding of nucleosomes to particular pieces of DNA, but it's not a specific sequence which they bind to. It's a structure that they bind to. So again, it's a lot like you think about some of the DNA repair proteins. They bind to structures. They don't bind to very specific sequences. And so here, you have to have this wrapped DNA that wraps around. Turns out some DNA is a little easier to wrap than other DNAs. And what that easier DNA to wrap is if you have these AT base pairs, which are in the minor groove on the inside of your nucleosome. And so basically what this means, if you have a stretch of about 150 nucleotides where you've got AT base pairing about every 10 nucleotides, because you remember it's 10 nucleotides per turn in your normal DNA, that particular 150 nucleotides is going to be better at binding nucleosomes than, say, a piece of DNA that doesn't have AT residues every 10 nucleotides. So you get a little bit of binding, but again, it's binding of a structure, not a particular sequence, which is what the nucleosome is interacting with. So core nucleosomes, 150 bases, but we've got also more DNA binding proteins, and in fact a whole bunch more DNA binding proteins that you find interacting with chromosomes. To start with beads on a string, you add histone H1. Histone H1s are a lot less well conserved than those core histones. So it turns out you've got lots of different histone H1s present around. But they're still present at very high concentrations. They have very high amounts, if anything, even higher amounts of positively charged amino acid side chains than the core histones. These guys interact with not only the DNA present in these nucleosomes, but also they interact with the tails on the core histones. Those core histones are going to interact with DNA beyond that particular histone. And basically what you end up with is you've gone from these bead in a string structure here to the structure that most of the DNA is in most of the time in your nucleus, the 30 nanometer filament. So that's basically diagrammed down here. lots of nucleosomes that interact with each other, and lots of other DNA binding proteins. They could be specific DNA binding proteins. They can be non-specific DNA binding proteins. We'll talk a lot about the specific DNA binding proteins after the midterm. So what do these nucleosomes and all these extra proteins look like at a higher resolution level? Well, the answer is we really don't know. But there are lots of models, and this is sort of the current model for how we think that these nucleosomes are interacting with each other. This is a cartoon of that 30 nanometer filament down here. There's the link that I had to YouTube that has the nice replication, transcription, translation. The beginning actually shows some really nice animations of these. But basically, what's happening is these nucleosomes are stacking down on top of each other, and stacking down on top of each other in such a way that the tails can actually reach out and interact with the next nucleosome, those histone tails, in order to help compact that particular structure. And so you can see that a little bit better if we now look at some of these tails. Here are those tails actually hanging off of the structure. So you think the nucleosome is kind of like this disc that you have present in the middle. These tails can be reaching out, and this one in particular can be reaching down to the next disc that you're going to have of the next nucleosome in your 30 nanometer filament. You can see that a little bit better here. Here we've got each of those histone tails reaching out, interacting with the next one of these particular nucleosomes. Also look at a little bit more detail at histone H1. This binds to a core nucleosome. You can see this is actually a dashed line here. That's because we don't actually have any kind of high-resolution structural data for a lot of histone H1, but at least this part we do. And we can see basically what this does is it pulls the two ends of the DNA that would be coming off of your 150 nucleotides that you have in the core nucleosome. That extra piece is just kind of pulled over even closer to the other end. And that way you're protecting a little bit more DNA from digestion by micrococcal nuclease, and you're compacting the DNA a little bit more. One other thing here is that this, you can see nicely from the crystal structure, is that histone H1 is not binding in a nice symmetric way like you'd have with all the core histones. It's an asymmetric binding that you have. It actually binds closer to normally the exiting DNA that's coming out of your nucleosome. So we've got our 30 nanometer filament, but 30 nanometer filament's all well and good, but it's nowhere near enough compaction to get everything inside the nucleus of a eukaryotic cell. So how do we go about getting more compaction? Well, basically what that seems to be is taking these 30 nanometer filaments, which are pretty much all histone proteins, those non-specific binding proteins, histones interacting with each other, and then binding them to yet another set of proteins. And these are the so-called scaffold proteins. And so that's just this red line down the middle here. These scaffolds are, again, they're proteins present inside the nucleus. They interact with these specific regions that you have in your DNA. These are your scaffold attachment regions. Again, molecular biologists often don't have much in the way of imagination, and they come up with these names. So the scaffold attachment region basically attaches two ends of a loop of this 30 nanometer DNA, and that attachment basically separates that piece of DNA from all the rest of the DNA. And we'll see that's really critical later on when we talk about regulation. It turns out that that loop of DNA is pretty separate from all of the other DNAs that you have in the cell. And that turns out to be really important for regulation and also how you have compacted or non-compacted chromatin. So these loop domains basically seem to associate and give these even larger helical structures. And again, this is quite nicely shown in that video, which I'll re-put the link back up again. Another way of looking at this process is here. Again, this is just a model for looking at these structures. We've got non-histone protein, scaffolding proteins down here, and the 30 nanometer chromatin filament, which is almost all histones, associated with that at these scaffold attachment regions in between. Now, these individual domains are also very often supercoiled, and you can see some writhe here, which is also allowing some extra compaction. And remember, if you fixed two ends of a piece of DNA relative to each other, that's a place where you can actually get supercoiling that happens between those attachment points. And so it's a nice way of getting this compaction taking on. You can actually visualize some of this. Here's a scaffold set of proteins down here. And if you look carefully, hopefully it shows up on this screen. I think it does. Each of these black lines up here represents the DNA. Here we've removed all of the histones, so gotten rid of them, and you can see that that's really given those histones give you a massive amount of compaction. All this gray stuff down here are just these individual filaments of DNA, which are all sort of crossed over each other. So it gives you an idea, again, what kind of compaction you're getting due to these particular histone proteins, but also clearly there are these scaffold proteins which these individual, again, usually 30 nanometer filament domains are attached to. So the next step, once we've got these 300 nanometer filaments, which again is pretty much all these things compacted next to each other, under certain parts of the cell cycle, you need to compact this part of the chromosome all the way down to a mitotic chromosome. And so again, this is the maximum amount of compaction that you get of your DNA under any system. And we've actually talked a little bit about how some of this compaction takes place because we talked about SMC proteins in bacteria. Because bacteria have exactly the same kind of problem. You need to compact your DNA. The DNA is too big to fit inside a bacterial cell. Here, DNA again is too big to be fit inside a eukaryotic nucleus. Of course, the nucleus breaks down when you have mitosis, but you still need to compact your chromosomes so that you can separate them nicely from each other. And it turns out that these are the SMC proteins, the structural maintenance of chromosome proteins. Here, so let me move this out of the way. This is the so-called Condensin protein, but it's got an almost identical structure. It's not surprising because the sequences, again, are quite similar. These SMC proteins to the muck proteins that we had already looked at in E. coli. They're long, anti-parallel coiled coil with ATP binding and hydrolysis domains and hinge domains that attach with each other. And these guys, just like the case in bacteria, basically sort of serve as clips. They clip around pieces of DNA and hold them together with each other. And you can see that down here in a cartoon form. Each of these guys has bound around two pieces of DNA. These head domains like to interact with each other. And so just by themselves, all of these SMC proteins interacting with each other, if they're clipped around DNA, they're going to pull that DNA together in sort of a helical form. Here it's shown as a circle, but usually these guys are stacking on top of each other as helices, and you can see that over on this side. Another way of looking at this is here, just a different image of basically the same thing. Again, coiled coil head domains that are interacting with DNA, these hinge domains which are just pulling together two chromatin fibers, and these two chromatin fibers could be beads on a string, they could be 30 nanometer filaments. It's just these condensins are binding around them and holding them together. There's another protein here in this particular case that helps bind these two together. Cohesin, I don't even know where that comes from. Anyone speak German? Kleister is German for glue. So sticking the two ends together here, it's a glue which is helping pull these two strands together. And again, it's just basically holding your DNA together. Here, also giving you binding to separate one particular loop of DNA, which again is usually a separate unit from all of the rest of your DNA. So we had Cohesin, which is helping to get these structures together. There's another set of SMC proteins which really helps you pull together metaphase chromosomes, and that's a protein called Condensin and then also Cohesin. So lots of these proteins, again, with these long coiled coil leucine zipper domains that they have in their proteins together with ATP binding and sticking together. Condensin goes together in the middle of these mitotic chromosomes. If you just look with an antibody to these proteins, you see them right in the middle with all these loops that have come out around them. If you look at a mitotic chromosome and also use an antibody to these proteins, you see they're right down the middle of these mitotic chromosomes. So they're clearly holding a lot of these loops together. Another one of these SMC proteins is something called Cohesin. And what it does is it holds sister chromatids together. I had some people in office hours I think last week asking me about how you get these chromosomes to line up with each other. Well, you can get your normal mitotic binding, crossing over taking place, but you have to have the two chromosomes relatively close to each other to have meiotic recombination, which is cutting and then homologous recombination that goes over. The way that that happens is through another one of these SMC proteins, another basically ring or loop protein, which now holds your two sister chromatids together. So again, it's their maternal and your paternal chromosomes that are being held together. Once they're held together, then you can have this meiotic recombination, which takes place between the two. Of course, once you've had metaphase, where you get these chromosomes together, you need to separate them relative to each other. And the only way to do that is you have to break the cohesin. And so the cohesin is broken through a proteolysis mechanism, and this is a really clear part of the end of the cell cycle. Once you finish mitosis, you need to break down that molecule that's holding the chromosomes together, and that's what allows the mitotic spindle machinery to pull your chromosomes apart. So this is what you've got. It's one of my favorite micrographs here. It's our color EM. No, it's a colorized EM picture because there's no color at that kind of wavelength. But here you have a compacted chromosome, completely compacted again, cohesin and condensin in the middle of it, telomeres at these ends, centromere right here, and that's a centromere where you're going to get the two things pulled apart. So before we start talking about hetero and euchromatin, any questions about sort of chromosome condensation per se? Yeah? Yeah. Yeah, so if you think about it, so I mentioned that all of these loops, they're very often supercoiled. And so dealing with supercoiling, you're going to have the topoisomerases. And not surprisingly, you have those topoisomerases also at the scaffolding right in the middle of your chromosome because they're helping with that supercoiling. Yeah? Mm-hmm. Right. So the question is, again, repeating so that this will be recorded, is differences between histones and nucleosomes. And so the nucleosome is the DNA plus those histones. So the nucleosome is that DNA protein complex. Okay, so let's move on and talk a little bit about this problem that I've mentioned a couple of times already. And that is that compacting your chromatin is all well and good if you're going to be doing separation, mitosis, etc. But a lot of the time, particularly in interphase, you need to be able to access that DNA. You need to be able to get specific DNA binding proteins. You need to be able to get your DNA polymerases, your RNA polymerases, all of your regulatory proteins associated with that DNA. And so to do that, you have to have a way of having DNA either be compacted or not compacted. And one way that people talk about this is in terms of open DNA or closed DNA. One way you can look at this is actually very similar to that micrococcal nuclease experiment that we talked about. If you now look at DNA, which is encoding one of the globin genes, globins are wonderful examples, mostly because it's easy to isolate blood cells that have these genes in them. So if you look at globin genes in either a cell which is actively expressing one of these globin genes, or here, a cell where you're not actively expressing these globin genes, you purify the DNA from those cells, you treat it with a nuclease, in this case it's DNase, and then look at that particular piece of DNA in a cell which is actively expressing this gene, it gets broken down. And so you just look at the DNA here. With DNA, gets broken down really quite quickly. On the other hand, at this highest amount of DNase, if you take these now from other cells that are not expressing the globin gene, you can see that there's no DNase digestion that happens there. You've got lots of this band here, which is from your southern blot, that you're looking at. So basically what that says is this DNA doesn't look like this DNA. This one is in our open format. This is in the closed format. So open and closed, we have to come up with some other names for this. They're also known as hetero and euchromatin. And so what we're looking at here, euchromatin is U being for true, like you have for a eukaryote, it's having a true nucleus. So euchromatin, this is your open, expressed, active chromatin. So we're not going to talk too much more about that. We'll talk much more about the heterochromatin. And heterochromatin is that chromatin which is not being expressed. It's much more resistant to gene expression, about 10% of it inside the cell. Right here, we've got some bar bodies. These are classic heterochromatin, heavily compacted DNA, and that compaction of the DNA leads to no expression in that particular case. There are two kinds of heterochromatin. Either heterochromatin that's always heterochromatin, always compacted. That's what's called constitutive heterochromatin. Centromeres and telomeres are really nice examples of that. Centromeres are always compacted. There's no gene expression that you get in centromeres. Telomeres, they're always compacted. There's no gene expression that you get in telomeres. On the other hand, facultative heterochromatin, which is the interesting kind, because that's the kind that can sometimes be compacted and sometimes not. We've already shown here, this is the bar body. The X chromosome is a really nice example of that. It's actually probably the best understood of these because you can actually see that you have compaction that happens here. And this is actually also visible under the light microscope, the so-called bar body. And for a long time, this was what was used, little topical stuff here, in the Olympics to determine if a male or female athlete, because there were some issues at some point about where they were. And so these bar bodies were an indication, because females are superior to males because they have two X chromosomes. One of those is inactivated, and that inactivation happens through this heterochromatin process. We know quite a lot about it, and we'll talk about this more when we talk about regulatory RNA a little bit later on in the course. But it's not just bar bodies. If you look at a normal cell, here is the nucleus, where we've got a number of areas of compacted chromatin which are present just in a regular nucleus. It's harder to see these kinds of things under regular microscopes. There's another way of looking at that. Here we have the euchromatin, which is basically the unfolded form, and about 10% of your chromatin is really heavily compacted. How does this happen? Well, we already talked about how you get chromosome compaction already when we talked about nucleosomes and 30-nanometer filaments, etc. Well, it turns out that it's all about the histones in terms of getting this compaction to take place, at least in terms of the facultative heterochromatin that you have normally inside the cell. What's happening? Well, you can get a little bit of an idea. If you just purify heterochromatin, you find that many of the histones that are associated with that heterochromatin are methylated. So histone H3 in this particular case, methylation at lysine 9, methylation at lysine 27. Lysines have what kind of charge? Positive charge. If you are now methylating those lysines, are they going to have a positive charge anymore? No, you're not going to have that positive. Basically, you've neutralized that positive charge here. You also can purify histones from euchromatin, and what you find here is that very often you have acetylated groups here, histone acetylation. You have phosphorylation. Phosphorylation is doing what? It's giving you a charge, but what kind of charge? Negative charge. What's that going to do relative to DNA? It's going to be pushing it away. So if you think about euchromatin, it's going to be less compacted, so you're getting that move away. Yeah?
A couple of questions. One, what is the 3 on the methyls?
So what's the 3 on the methyls there? We'll get back to this a little bit later on, but that's a trimethyl. So actually 3 methyl groups that are added to your lysine.
Okay, and I was wondering how the methylation would stop it from gaining a positive charge if it's methylating the amine.
On that particular, we'll look at that. We'll see actually some of those. Trimethylation, yes, 3 methyl groups, not just and you can get monomethylation and dimethylation as well, and that doesn't change the charge. So yeah, was that the only question, or you have another question? Okay, yeah. I don't think I have any images of, because we actually have some really nice structures, so we know exactly where those methylations are taking place. I don't think I have those in the lecture notes, but I can send those to you. So. These are just some of the modifications that you find, again, particularly if you purify histones from either heterochromatin or euchromatin. But it turns out that these are only a very few of the modifications that you can get. So if you look at all of the core histones, and again, these are the tails of the core histones. Here's the N-terminal tail of histone H2A, number of potential modifications. These are potential modifications. They rarely all happen at the same time. H2B, probably histone H3 is the best studied, partly because it seems to have the most modifications that take place with it. So here's an example, and particularly this lysine at position 9. It can be methylated, it can be acetylated. You can get 1, 2, or 3 methyl groups that actually get added to it. But you have a number of other modifications that can happen, and again, very rare these are happening at the same time. Phosphorylation, etc. And that's only one, let's say, phosphorylation, acetylation, and methylation, only 3 of the possible modifications that you can have. Turns out you also can have ubiquitination, and that's just adding the ubiquitin molecule to your histone. Remember, ubiquitin is actually a peptide, it's almost 80 amino acids. So it's a pretty major modification that you have that happens to your histone. And this is a histone modification that doesn't lead to degradation of the histones. It just seems to be a signaling modification that happens. There's also another molecule called a small ubiquitin-like modifier, also known as SUMO, which again can modify a number of these histone side chains. And so really covalent modification of histone tails seems to be really critical in terms of getting changes that happen with compaction of your DNA, which is going to heterochromatin, versus the opening up the euchromatin that you have of active DNAs. And this is just another way of showing this again. Histone H3, acetylated, methylated, phosphorylated, etc. So there are lots of different things that can happen, different modifications that can happen, and it's those combinations of the modifications are what people call the histone code. And so if you have trimethylation at lysine 9 and trimethylation at lysine 27 in histone H3, that seems to mean heterochromatin. And so we'll look more at that in just a second. But in addition to having these modified tails, there are also what are known as alternative histones. They're present at much smaller amounts that you have inside the cell, but the key to all of these guys is that they all have the histone fold, which are those 3 alpha helices that can interact with each other and then dimerize with other core histones. These guys are actually going to now be in the nucleosome. And they were asking about the nucleosome a second ago. So the nucleosome is the histones plus the DNA. These guys get into the nucleosome and cause that nucleosome to have a slightly different function. 2 probably best known of these are modifications of histone H3. This histone H3, again, is the one which gets heavily modified. Here, histone H3.3 seems to be present in areas of the genome where you get lots of transcription. And then at the opposite end, this histone H3-like protein, SYN-PA, is what's present at the centromere. Again, SYN for centromere. And this centromere, talked about, that's some of that areas of the constitutive heterochromatin. So always compacted. And so never going to be activated. And what you can hopefully see here, this blue is a slightly different color than that blue. That's because this doesn't have the same sequence at all in that tail structure. So you can't do all of these extra modifications which are happening with that. So what is this histone code? And basically, not so much what's the histone code mean. We can look at these things and say, oh, we get heterochromatin, it means this. We get euchromatin, means that. But how does a cell know that we've got any particular kinds of modifications? So we mentioned these 5 major modifications before: acetylation, phosphorylation, methylation, ubiquitination, and sumolation. We're going to concentrate on acetylation and methylation here. Already mentioned, methylation is very common in regions of heterochromatin. So the way that I like to think about this is there are these so-called chromodomains, and so chromo for heterochromatin, which recognizes very specifically histone methylation. And so any protein that has a chromodomain that's associated with it is going to recognize methylated histones. Those methylated histones are usually associated with heterochromatin. On the flip side, if you look at acetylation, which we haven't talked about that much, but that's basically what's leading to opening up of your heterochromatin into euchromatin, these bind to the so-called bromodomain proteins, and these recognize acetylated histones. Phosphorylation, ubiquitination, we're not going to concentrate on that much. So let's look at a couple of examples of this. Here, I already talked about methylation at K9. It's usually met3. That's what they should have on here. There should be 3 methyl groups. Very common in heterochromatin formation. On the other hand, acetylation at position 9, but curiously enough, usually in combination with methylation at lysine 4, leads to gene expression. Acetylation at 14 and 10, and phosphorylation at serine 10 lead to gene activation, etc. And so what it basically means is we've got lots of different modifications, and it's going to be those combinations of the modifications which are going to lead to the readout of that particular histone code. So we'll look at 2 examples of that right now. The first one is going to be where we have methylation. And again, this is methylation at the lysine at position 9 of histone H3. You have a specific protein, histone H3K9 methyltransferase. What does it do? It methylates that particular position. Once you've had methylation at that particular position, and again, this is trimethylation, now you have binding of this heterochromatic protein 1, HP1. As soon as that binds to this methylated histone, HP1 loves to bind to itself. And so basically it's that HP1 interaction with HP1 which leads to the formation of your heterochromatin. So here, HP1 binds to itself, forms your heterochromatin. Now what's interesting is that if you had all this heterochromatin forming all the time, and it turns out that there's a feedback loop that goes with this. Once you have methylation, you get more methylation. You end up with spread of heterochromatin. And spread of heterochromatin can be a big problem because you might have some regions of the genome you really want to express. And so we use that example of the globin genes. So globin genes you want to be nicely expressed in cells that need to be producing hemoglobin. Well, you want to have them turned off in cells that are not making hemoglobin. So histone H3K9 methyltransferase is going to be great in those cells where you don't want globin gene to compact that part. But if you've got cells that don't have that and need to be actually functional, you're going to want to acetylate these histones. And so this is a really basic way of looking at that. But as always in biology, it's more complicated than that. So it turns out that what we have is multiple combinations of these binding proteins and modification proteins. And very often, these are going to be starting out with one specific gene regulatory protein. So in this case, we've got a gene regulatory protein that interacts with something like that histone H3 K9 methyltransferase. So the regulatory protein binds to a specific piece of DNA under specific conditions, cause a modification of the histone. Once that modification has been made, then you have these code reader, code writer proteins, which will bind to that modification, but also bind to your writer protein and lead to multiple modifications that happen in that region of the DNA, which is near to where you had this gene regulatory protein. So it's basically a positive feedback loop. You have one event that takes place, again, binding or activation of this one regulatory protein that then leads to multiple changes that happen right there. Of course, again, it's not just those reader-writer complexes. We also have these so-called chromatin remodeling proteins, which move histones around, move histones forward, move histones backward. These are ATP-dependent proteins, so you actually do need to use ATP to move nucleosomes around. And they basically help these reading and writing complexes in order to get a modification that happens in one place to spread from that one position. Now, of course, spreading, as I mentioned before, is a real problem. So if you've got modification at one point, it's a positive feedback loop. You're just going to keep going, and potentially you're going to modify all the histones in your DNA, and then everything's going to be silenced. Well, you need to have some of the genes expressed some of the time. So if you have these feedback loops that are leading to lots of modifications, and you can think about the modifications based on those last two slides and just flip them over and say, okay, we're going to have activation of chromatin. So instead of methylation of your histones, you can have acetylation, which leads to activation. Same thing can happen there. But you have to have these barrier proteins, because if you didn't have barrier proteins, anywhere where you had heterochromatin formation or euchromatin formation by these reader-writer complexes and chromatin remodeling complexes, that's going to spread through your whole chromosome. So you want to have these regulated somehow, and one of the ways that that happens is through the binding of these scaffolding proteins. You remember at the bottom of these loops of DNA, you've got scaffolding proteins that are bound to your DNA. And that blocks the progression of these histone modifications as they're moving along through your genome. You can also have specific proteins that will block that. Most of this, again, happens to be structural. It can be bound to the nuclear pore, barrier proteins. Or sometimes you actually even have an enzyme and some enzymatic activity that will actually act against what was happening there. And so if you think about the case that we had with histone methylation, if you have a histone demethylase that happens to be bound to one particular place in the DNA, that would stop the progression of this wave of heterochromatin formation. If you're talking about acetylation, you have the same kind of thing which is happening there. I just want to finish up just reminding you that we've got alternative histones as well as these modifications. How you get alternative histones associated with DNA, we can talk about offline if you're interested in that. But in this particular case, you can see nicely that these alternative histones, particularly the SENPA1, which associate with the DNA present at the centromere, and that particular tail, which you remember for SENPA is actually different than your normal histone H3 tail, that interacts with the cell division and particularly the chromosome separating machinery. So the tails here now interact with this kinetochore and eventually microtubules that will pull your two chromosomes apart. So with that, we'll stop before talking about telomeres. We will talk about telomeres after the exam, and so those will be on the final.